There's something that many persons fail to realize as it relates to the ABO blood group system in terms of the molecular genetics. Some persons believe that there is only one ABO allele, but since cloning was taking place in the 1990s, the complementary DNA corresponding to the messenger RNA transcribed as the blood group ABO loci, it was determined that more than 250, yes, you heard that right, there are more than 250 ABO alleles that have been identified by molecular investigation. And the ABO genes is located, as we had disclosed before, at chromosome 9. And in this area, it consists of 7 exons. The last 2 exons, the 6 and 7 exons, they encode for the catalyst or the catalytic dominance of the ABO glycosyl transferase. So exon 6 and 7 actually constitute 77% of the gene, which most of the coding sequence is lined in number 7, right? So amino acid substitution result primarily from the deletion, the mutation, or gene recombinations within these 2 exons of coding DNA. So from this, we can derive variant ABO glycosyl transferase, which are responsible for less effective transfer, or they react weaker when the conventional sera is used. Anti-sera, anti-A, anti-B is used in identifying the blood group. The ones that we need to bear in mind, the 7 that we need to bear in mind is of the common ABO alleles includes the A1. Yes, A1. Remember, A1 is the one that we use in the reverse blood grouping. Then we have A1 variant, which is A subscript 1 and V. And with that in mind, let me just put that on the screen so that we can all have a better visual. So the 7, I'm going to put it there, 7, S-E-V-E-N, the 7 alleles that we need to be mindful of, all right? And these are the ABO alleles. So A-L-L-E-L-E-S, all right? So number 1, we're talking about the A1, and that is the A1. Then we have number 2, the A1V, which is the A1 variant. All right, and remember we said we have A1 and we have A2. So in your mind, I want you to think about having an A2. So you have A1, A2. So you're saying to yourself, is there an A3? Actually, there is an A3, but it's not a part of this the 7 allele. The next one, number 4, is going to be the B1. Then we have 5 is the O1. Yes, and 6, guess what? We have a variant again. So it's going to be O1V because it is the O1 variant. All right. So this is O1 variant. Then number 7 is the O2. So yes, we have A2 and we have O2. So this is just something for you to be able to mentally visualize that these 7 ABO alleles do exist. And as we will come to note, the A portion has also many variants. But if you divulge a little bit into molecular genetics, which is a part of the whole systematic training in becoming a medical technologist, a laboratory medicine practitioner, a clinical laboratory scientist, medical laboratory scientist, whichever route you decide, then you will be given some idea as to how molecular genetics plays an important role in the ABO blood group system. So if we are focusing in that area, we can go a little deeper. But for this study, for this moment in time, we will leave that detail for another sitting. So I want you to understand that all ABO antigens arise from mutation in the single ABO gene. Only 3 specific mutations show a higher frequency in the population, indirectly leads to the change in the epitope structure, resulting in A, B, or O specificities. All right. So there are just 3 mutations that occur that has been noted to change your blood group. So somebody can be A and they present as B. All right. And somebody can be A and then change over to B. So when this happens, this is determined to be an incompatibility issue that is taking place, and then we as a clinical laboratory practitioners have to decide how do we go about resolving these anomalies, these discrepancies in the ABO. So the two mutation of substitution changes the specificity of the enzyme for the alpha N acetylgalactosamine transferase. All right, and this is the enzyme for the A, or the A enzyme. All right, and then it would convert from that to an alpha 3D galactosyl transferase, which is the B enzyme. The A, more difficult to call, the B, a little easier to call. All right. So these glycosyl transferase in turn introduce an alpha 1,3 N acetylamine, which is the A, or it can also introduce alpha 1,3 galactose, which is the B carbohydrate at the end of the H oligosaccharide or the glycan chain, right? The third mutation can be a deletion within the 5' region. And you know that when we're doing molecular genetics, we have the prime region, we have the folding, we have the replication process of the DNA and the RNA taking place. So this is the one of the 5' location or region that a mutation can occur, and this mutation at the 5' is usually a deletion. So a deletion within the 5' region of the catalytic dominant creates a premature stop to the codon that inactivates the enzymes altogether, leaving the H glycan unmodified. So you have H that are unmodified, right? So these different glycan differentiate the A, the B, and the O antigen or the epitope. So when we have this situation, we realize that we can have discrepancies in our interpretation of the blood group system. Now, let us try to wrap our minds around the formation of the A, B, and H soluble antigens. So we already discussed the A, B, and H antigen as a whole. Now let us talk in more detail about the formation. So the ABH antigens, these are integral part of the cell membrane, especially the red blood cell, the endothelial cell, platelets, lymphocytes, and the epithelial cell, just to name a few. The ABH soluble antigen can also be found in all body secretion. And you may be aware that we spoke about the SE, which means secretor, and then we spoke about when we talk about secretion, we're thinking about things that are displayed in other areas. All right, so there are different areas in which secretion occurs from the body. You have the sweat, you have the saliva, you have the all right, we're gonna get into that in a little while. So I just want you to just wrap your mind around that idea that your ABO blood group system, in part, can be identified based on secretions. All right, so let us go back now to the ABH soluble antigen. So the ABH soluble antigen can also be found in all body secretion, right? Just remember that. So their presence is dependent though on the ABO genes inherited and on the inheritance of another set of gene called the SE, which is the secretor genes. And these genes regulate their formation. So even though we have the ABO gene, we need to have the secretor gene activated so that we can have the secretion being easily identifiable in our body fluids. All right, which are easily accessible to the majority. All right, beautiful. So we are getting a clear understanding. What we should note that 8% of random US population are known as secretors because they have inherited the capital S, lowercase e, capital S, lowercase e, or capital S, lowercase e, lowercase s, lowercase e. All right, let me just put that secretor genes. Comes to us from the SE, S, let me, SE, SE, all right? So this is the genes that is necessary. In order to be a secretor, you can all either have SE, capital E, common, capital S, common E. Then we have capital S, common E. So with this, you have the secretor, number 2. You can also have the secretor with capital S, lowercase e, lowercase s, lowercase e. All right? So the inheritance of the SE gene, we spoke about this, the FU, T2. This here is the SE gene is coded for. So the inheritance of the FUT2, which is the secretor genes, codes for the production of transferase. And the transferase that we're talking about is the alpha 2L, and it is fructosyl transferase. FUCOSYLTRANSFERASE. All right, and that is used as a modifier. It modifies the type 1 precursor in the secretion of the H substance, and we know that H substance is necessary to determine your ABO ABH blood group system. All right, it can then be further modified to express A and B substances in the secretion. And when we think about secretion, the first thing that's supposed to come to our mind is saliva, because in the mouth we have amylase, we have secretors having their blood group easily being determined just by a buccal secretion assessment. All right, so we are thinking along those lines. So a group A individual who is a secretor will secrete glycoprotein carrying the A and H antigen. So if you are a group A, you will carry, based on your secretion protocol, so SESE or SESE, you will secrete the glycoprotein and this glycoprotein that you will secrete carries the A and the H antigen. So now you can see why you use anti-A, anti-B, anti-H in our determination of blood grouping system. All right, so that is just something for us to think about. So but what if the person is B? Let's go to that. So the person is group B. We're going to expect them to have the secretor genes as well, SESE and SESE, right? And they're going to still have their glycoprotein and then they are going to be giving us a direction towards the secretion of the B and the H antigen. All right, it is the presence of the SE gene specified let's say alpha 2L fructosyl transferase that determines whether an ABH soluble substance will be secreted. So people who inherit the SE, we need to talk about this. People who inherit the SE, these are lowercase So persons who inherit the SE genotype are termed non-secretor. So if you inherit those genes, you are a non-secretor. And that is just how it is. So some persons are secretors, other persons are not secretors. So what I want us to take this time to do is to just go over some of the differences between the ABH antigen on the red blood cell and the ABH soluble solution or soluble substances. All right, so let us gear our minds. Let me try to do a division on this board so that we can all see. So we're going to talk about the ABH antigen on RBC. All right, and then we're also going to be talking about the ABH soluble SOLUBLE substances. All right, so the ABH antigen on the red blood cell. First thing we need to remember is that the red blood cell antigen can be a glycolipid, it can be a glycoprotein, it can be a glycophospholipid. All right, so we have three, 1, 2, 3. So it can be a glycoprotein, glycoprotein, glycolipid, glycolipid. It can be a glycosph sphingolipid, lipid. All right, but for the secretion, the ABH secreted soluble or ABH soluble substances, secreted substances are only glycoprotein. All right, glycoprotein. So they are only glycoproteins. So that is one distinction that we have already. So the next thing that we want to remember is that for the antigen, the red blood cell antigen are synthesized only on type 2 precursor chain. So synthesize SYNTHESIZ on only type 2 precursor chain. All right, the next thing, if that is on type 2 and we talk about type 1, I want you to put in your mind, if this is type 2, the other one must be type 1. All right, so for the A, B, and H soluble substances, secreted substances are primarily synthesized on type 1 precursor chain. All right, so synthesize SYNTHESized on type 1 precursor chain. All right, now let us think about it. We had discussed this already, that the type 2 chain refers to the beta 1, which has 4 linkages, in which the number 1 carbon of the galactose is attached to the number 4 carbon of the N-acetyl glucosamine sugar of the precursor substance. All right, sorry about that. All right, so we have type 2, and it's the same thing that we have been discussing before. So type 2 goes to the beta 1, and this is linked to 4. It has 4 linkage, Linkag, right? That's what we need to understand. So 1 is linked to 4. Now, for the type 1, which is on the AB and H soluble substances, we have type TYPE1 chain, all right? And then, as we stated, referred to as the beta 1, and that is linked to number 3, or it is 3 linkage, all right? So for the A, B, and H, we have 1 to 3, and the 3 is a linkage in which the number 1 carbon on the galactose is attached to the number 3 carbon on the N-acetyl glucosamine sugar of the precursor substance. All right, now let's move right back to the ABH antigen on the red blood cell. What I want you to bear in mind is that the enzyme produced by the H is FUT1. The gene is alpha 2L fructosyl transferase, which acts primarily on type 2 chain, which are prevalent on the red blood cell membrane. All right, so remember the enzyme. So when we talk about ABH antigen, we're talking about enzyme ANZYME produced by H is FUT1. All right, just remember that. And for the AB and H soluble substances, the enzyme produced by the secretors, so enzyme produced by secretors, and the secretors are the SE, and we know that this SE is going to be FUT2. All right, that is a gene, and this gene is still, we're still talking about the alpha 2L fructosyl transferase. All right, its preference is towards action on the type 1 chain in the secretory tissue. So if we bear that in mind, we are on good terms in understanding the concepts of these ABO system, not to mention the ABH antigens on the red blood cell. Then we have the ABH soluble substances, which when we talk about soluble substance, we're talking about the substances that are secreted. So this here gives us an idea that the ABO blood group system is not as basic as many persons may consider it to be, but it has some complexity within it. However, it is the basis on which the modern blood transfusion system is based. So let us just talk about in brief the idea that ABO has subgroups. And the original report of most ABO subgroups were made before the availability of the monoclonal typing reagent currently used in routine ABO grouping. So the ABO subgroup represents the phenotypes showing weaker and variable serological reaction within the commonly used human polyclonal anti-A and anti-B, not to mention, and anti-AB reagents. So some labs use A and B only, while other labs use A, B, and AB. All right. So the AB is usually used to confirm the A or the B and to find out if there are any variants in the whole system. All right. So what I want us to do in our next sitting is to go into some details about the A subgroup, because yes, we have A1, we spoke about that. We have A2, we spoke about that. We mentioned A3, but there are other A's that we could investigate. And I want you to be on the journey as we investigate those A subgroups. But before we delve into that, I want you to remember secretion and the secreted genes, SE, and also the A, B, and H substances. All right? So when we're talking about body and body fluid, and we're thinking about the ABH substances, I want you to understand that they can be detected in saliva, they can be detected in tears when you cry, they can be detected in your urine when you urinate, they can be detected in the digestive tract when the food is being digested, they can be detected in the bile coming from the bile duct, they can be detected in the milk because the milk in lactation for mammals is a secretion. They can be detected in the amniotic fluid, which is the area in which house the infant. They can be detected in pathological fluids such as the pleural fluid, the peritoneal fluid, the pericardial fluid. It can be also detected in ovarian cysts. Thank you for spending some quality time with us on the Professor Kareem Ainsley Encounter. As you always know, when we come to this period of time, we say class is dismissed. Have yourself a great day.